Okay, great. Thank you. Welcome back. For those of you who don't know me, I am Kiki Syrad. I'm the Director of Research and Innovation at Great Ormond Street Hospital, and it's a real pleasure to be here. We had excellent sessions before the break, and there's a lot more of that to come. And as you can see, the BRC really impacts patient care and outcomes, and we will have more lovely examples of that this afternoon. So I have the pleasure now of introducing Professor Paul Gisson, who will talk us through the gene, stem and cellular therapies theme. Paul is a clinical professor of pediatric metabolic medicine at the UCL Great Ormond Street Institute of Child Health. He is also honorary consultant in pediatric metabolic diseases at Great Ormond Street, and he also leads the gene, stem and cellular therapies theme of the BRC. So thank you, Paul.
Thank you very much, Kiki, and it's a great pleasure to speak to you today on behalf of our theme and my colleagues, Claire and David, who are deputy leads, and Alice and Havinder, and also Azizat, who is our theme manager. So it's, as I said, it's a great pleasure, but really this is just a preamble to the real stars that are going to speak after me. This is what our theme does, and we develop gene therapies to treat disorders, particularly those patients that are the sickest ones and they don't have therapies that are already available for them. So these are the most important ones. Our aims are to expand the number of clinical trials and basically make gene therapy accessible for more patients and with more different diseases. We also want to make trials safer and the children better. Our priority is to focus on the, as I said, the most severe diseases where treatments are not available. So just to mention a couple of recent achievements, the first one I'm going to talk about is the achievement of Claire Booth and her team, and also international collaborators. So the picture bottom right, which was not the favorite picture of Claire's, but unfortunately we couldn't change it. But essentially, that shows how the gene and cell therapy is done. As opposed to the traditional bone marrow transplantation, you don't need a donor, and the cells are taken from a patient. Then the gene therapy, so the vector with the gene in it, is put into these cells. The cells are expanded and then put back into the patient. So that really should make this treatment a lot safer and potentially also more efficacious. So Claire works with patients with immunodeficiencies, and in this particular disease called LAD1, which is an immunodeficiency, they've shown that the long-term follow-up, the 7 years or more, this therapy compared to the standard of care was safer and more effective for these children. The other achievement I'm going to talk about is the achievement of the team involving Sarah Garashian and Percy Zamrolia from GOSH. And they develop CAR T cells. So you see the picture of the CAR T cells. And similar to Claire's work, they take the T cells, these immune cells inside our blood, and they weaponize it with the gene therapy to make it targeting the tumors inside the patients' bodies. So whilst it was initially developed for the children, actually the company, which was a UCL spin-out company, took that therapy and then targeted the adults and performed the trials in the adults. And this treatment now is approved and is successful, effective, and safe than the standard of care in adult patients. Of course, Sarah and Persis are continuing to develop that for children, and these trials are continued, and hopefully this will also happen for pediatric indications as well. So our current project focusing on our GMP facility and making more products for gene therapy trials, both at GOSH and nationally and also internationally. We also develop therapies preclinically that can be taken on the translational pathway towards clinical trials. We work on developing more gene therapies for many more diseases in different organs, kidney, liver, brain. We are only one of the 2 centers, and that I did see Alex sitting here. We're only 2 centers in the world which performs the thymus transplants. Of course, we study the effects of the therapies on patients in the long term. In the following years, we'll continue doing this outstanding work and also develop the world-leading young scientists developing the gene therapies, but also educating the general public, children who are present here too, and also the clinicians and our colleagues about gene therapies, and of course always looking for collaborations in the field of gene therapy, expanding our target diseases. So finally, I will introduce our real new stars. One of them that's going to speak first is Rebecca, who is a PhD student in Birmingham, and her project is on safety of gene therapies in terms of the liver complications, and then Julian, who's leading the clinical trial of the novel therapy, potentially curative for the currently lethal condition. So over to Rebecca.
Thank you for the introduction. And as Paul's mentioned, gene therapies are an incredibly exciting and long-awaited treatment modality for patients who have historically had few treatment options. With new therapies, we do sometimes see new side effects. And as someone who's hoping to specialize in looking after children with liver disease in the future, my PhD research is focused on a new form of liver injury that we're seeing in patients after they're given gene therapy. As a bit of background, liver side effects after gene therapy are fairly common. In most cases, they're mild, and we only notice them when we do blood tests after a patient receives gene therapy. In some rare cases, the injury to the liver can be so overwhelming that the liver, which is normally a very resilient organ, becomes unable to maintain its normal functions. We call this liver failure, and this is a very serious condition. We don't fully understand the liver side effects and why it's particularly severe in some people, but we think it's due to the body's immune system fighting adeno-associated virus, which is a delivery vehicle that we use in some gene therapies that are administered directly into the vein. Sorry, that was an unexpected color. Before I embarked on my PhD project, I went away and looked at gene therapy trials that had been published in the previous 5 years. I wanted to see how these trials managed immune side effects in their patients. The major takeaway from that was that actually practice is very variable, and we don't really know what the best approach is. And this is at least partly because we don't fully understand why this liver injury happens. We also know from talking to patients and their families that they would like more information about the liver side effects because that would help them make a more informed decision about their treatment. So to study this fairly new clinical problem, I developed a project that ties together expertise from 3 different centers in the UK. I'm very grateful to be funded by the Great Ormond Street Hospital BRC, and as we've heard, the gene therapy expertise here is phenomenal. I've been around UCL for about 11 years, but for my PhD, I moved to Birmingham to work with the pediatric liver team in Birmingham Children's. This is a highly specialized service and one of 3 centers in the UK that look after children with liver disease. And finally, my main research is based in the liver labs at the University of Birmingham. So this is a well-known institute for liver immunology research. And what's really important for my project is that there's a very well-established infrastructure here for obtaining liver tissue that would otherwise be thrown away during transplant operations to use for research with patient consent. So the access to having liver tissue from humans is incredibly important to my project because we know that the side effects that we see in humans are not completely predicted by studies in monkeys, in dogs, in pigs, in rats, or mice. So having human liver tissue and human liver cells are particularly useful for us. Depending on my experiment, I can look at various things in the materials that we have. So for instance, in some experiments, I might look at the picture in pink, the structure of the liver. In other experiments, I might look at the very specific way that liver cells are organized into zones in the liver. Alternatively, I might look at the expression of receptors in the liver that might influence how the liver cells interact with the gene therapy, or I might be interested in the blood vessels in the liver. Overall, we hope that this project will help to clarify some of the mechanisms that lead to liver injury so that we know how to better manage it when we see it in our patients. We're very excited to have set up a new gene therapy program of work in our institute in Birmingham, and we're very grateful to the clinicians and the patients and scientists from multiple centres across the UK who have been very generous and very enthusiastic about pooling samples and expertise and experience. And ultimately, we hope this collaborative effort will help make gene therapies as safe as possible for those who need it. Thank you.
Good afternoon. I'm Julian Baruto. I'm very pleased to present you this gene therapy trial, which is testing a novel gene therapy technology, and the first baby having dosed at Gosh last year and have been successfully treated. So this baby had a liver genetic disease. This is a large group of conditions, around 150 conditions, and some of them can be really severe, requiring in the first months or years of life liver transplantation. And unfortunately for the babies and young children, the current gene therapy vectors technologies are not suitable to enable a long-term correction of the disease symptoms. This is because these vector tools, essentially adeno-associated viral vectors, are in fact not integrating the therapeutic gene. You can see in pink, this gene is not inserted in the chromosomes, and during the rapid liver growth and cell division at that age, you can gradually dilute the effect of your gene therapy, and in fact you just see a very transient correction of your disease. And because of the immune reactions caused by the first injection of the gene therapy system, you can't re-inject, and these children will not have a sustained correction. So this new technology is bringing a novel approach with gene insertion, in which the gene will be directly inserted in the chromosomes. And you can see that over time, the aim is that during liver growth and cell division, this therapeutic transgene will be passed to daughter cells and will enable long-term correction. The concept of the technology is based on two different viral-derived vectors, two AAV vectors. One which will be encoding for a nuclease, so a molecular scissor which will cut at a very specific and a very safe locus in the genome. And the second AAV will bring the therapeutic transgene. And these two mixture of different AAV will be infused intravenously in the patient. And the AAV vector is designed to have a high liver tropism and effectively insert this therapeutic gene in the liver cells. So the objective of this first-in-human trial was to see whether this technology will be safe in babies and could bring a long-term correction in these children. So the name of the clinical trial is OTC Hope. This is a first-in-human phase 1/2 clinical trial, which is targeting a severe liver genetic disease called ornithine transcarbamylase deficiency. And within this genetic disease, the most severe presentation, which is seen in newborn males with neonatal onset. So the baby was enrolled at Gosh, was a child who had presented in the first week of life a neonatal coma with a very high level of ammonia, so life-threatening episodes of metabolic decompensation, who had to be admitted in intensive care units to restore some life-compatible ammonia levels, and being started on the usual standard of care, which combines medications and diet. And these children, despite this treatment, had a second episode of hyperammonemic decompensation at the age of 3 months, and was finally dosed at the age of 6 months. 1 month later, he presented liver inflammation, what Rebecca mentioned earlier on, which was easily managed with some immunosuppression drug. And we were able to rapidly stop medications and diet, and the patient has been free of any medications and diet for more than a year now. So a really successful response to this new gene therapy approach. I would like to highlight the fact that it has been a huge teamwork involving many teams and many clinical units. And for example, on the dosing day of this patient for this gene therapy infusions, we had multiple people involved, and to get a successful procedure, we had more than 20 people who looked after these patients in some ways on that day. So this, to conclude, this is the first child dose with this new gene insertion approach worldwide. This brings an efficacy we have seen so far equivalent to a liver transplantation, but after a 2 hours infusion of a 10 ml syringe. And this really opens new opportunities for babies with severe liver genetic diseases. This is the picture of this child who made the headlines in the news earlier this year. And I really thank this child, the parents, and all the team involved, and thank you for your attention.
Thank you. Really very interesting talks. Thanks. So do we have questions in the room or online?
Okay, so to kick off, Paul, gene therapies are eye-wateringly expensive. Can you see a future where they will be cost-effective?
Yeah, this is a difficult question, and many gene therapies are expensive. So I guess in the future, so what we would like to do is to make it less expensive by, first of all, improving the cost of manufacturing, but also in parallel, there is a possibility of improving the cost of development the drug, because compared to the conventional drug development of gene therapy shouldn't take as long, and there are other pathways that are being explored. People talk about platform technology and approval of the platform as opposed to individual drugs that could potentially make it cheaper. So if you develop the drug cheaper, if potentially develop it in a different way using not-for-profit organizations, then that is possible to make these things cheaper on the big scale in terms of for the NHS to make it cheaper. Yes, there are ways of doing it, and we are going towards that.
Thank you. It's definitely one of the challenges. Other questions?
Thanks for a great session. Rebecca, it's a question for you. Are you looking at, so I work in neuroscience, and I think even for some of our CNS-delivered gene therapies, we still see some liver side effects in some of our patients. Are you going to look at that as well, or are you just looking at systemically delivered gene therapies?
So at the moment, we're just focusing on using ex vivo tissue will introduce the vector into the liver, so we'll be studying ex vivo. But from patient samples that we're collecting, the main samples that we have now are from children who've received gene therapy for spinal muscular atrophy, so intravenous delivery. And this is partly because of the access of liver tissue. It's available just for some of the conditions from either biopsies or post-mortem samples. If we are able to get liver tissue from other conditions, then we look into it. But for now, the main focus is intravenous delivery.
And I guess just a follow-up question is, do you think there's something inherent about, do you think certain people get that side effect because of something inherent in them? Or do you think it's a kind of class effect that anybody can get?
Great question. I think there's probably patient-related factors, so genetic predisposition. Sometimes HLA polymorphisms can also play a role. People have suggested also genetic polymorphisms in some of the interleukin genes, for example, might play a role, but also could be vector-related factors. So between different gene therapies, as far as I understand, there can be a difference in the proportion of empty capsids, for instance, or other nucleic acid contaminants. And probably both, all of it all plays a role.
Thank you. Thomas?
Julian, can I ask you to speculate how many of the genetic liver diseases might be amenable to that sort of knock-in treatment, say, over the next 5 to 10 years? Not 150, but realistically.
Speculation is always a difficult exercise. This is a platform, so the therapeutic gene can be adjusted and changed according to the disease of interest. I think in theory that could apply to most of these conditions. You need to have the therapeutic gene being able to fit within the packaging size of the AAV vector. So for some of these conditions that will not be possible, but I guess for most of them that will be the case.
And as a follow-on, it might also target some of the non-lethal diseases like hemophilia, for example, where you just need to produce something, and if you target them early.
Yes, absolutely. Phenylketonuria, all sort of more frequent conditions could be amenable to that. I didn't mention it in the talk, but the safe locus of insertion is in a gene which is involved in hypercholesterolemia, PCSK9. So in theory, we are disrupting this gene because the therapeutic gene is inserted within the middle of this PCSK9 gene. So they get double benefit in some ways. They have a cure of their disease potentially, and they have as well a reduced level of cholesterol, which can always be a benefit long term.
Thank you. Sergi.
For Julian, as you require integrations, at this point, how do you balance the level or amount of integrations so that the therapy does not dilute over time with the risk of these integrations? Is there a science or is it an art at this point?
This is a very good question. There is within the 6 months interventional trial, there is a liver biopsy which is planned at 6 months. And so the levels have the results have been analyzed but not publicly disclosed yet. But this is really a very precise system which enables a very limited off-target efficiency. But in terms of integration on-site one, this seems to be very good as well because so far we don't have these publicly available results for the liver biopsy, but we can see the PCSK9 protein, for example, in plasma, which is reduced by around 1/3. So clearly there was a cut which happened nicely there. And clearly we can see the outcome clinically for the child. So we can see that this has definitely worked in some ways.
Thank you. David?
Yeah, there's a question directed to Rebecca, but I think it applies to much of our work. Patients would like more information on liver side effects. Have you explored how to do this or how to address this need for more information?
To address the need for more information on the liver side effects, I guess hopefully my project will help with that. I know there are other people in UCL also looking at it, so we collaborate with Leanne and Judy Brewer, and they're virologists, and so they focus on a slightly different aspect, but we work together. My understanding is that because the liver side effects weren't necessarily predicted by preclinical animal studies, a lot of people are now revisiting older literature and older experiments to reevaluate what we know in the context of what we've seen with more gene therapies becoming available and thus more side effects being seen. And I think more people are looking into it, so hopefully we'll have more information. I will hand over to...
I think the information, so it's also important to realize that the majority of patients that received AAV gene therapies don't have significant, certainly no significant liver symptoms. So yes, there are occasionally, very occasionally, and these cases are quite high profile, the ones that develop liver failure even. Some conditions are more likely to, some patients are more likely to get them, and maybe because there is some underlying liver disease. But on the whole, AAV is safe.
Thank you. Maybe one more question and then we need to move on.
Okay, Julian, was everything ready beforehand, i.e. in vitro proof of principle? And were you waiting for a patient, or was your therapy rapidly developed once that patient was born and identified?
So we were contacted by a company, Ayukyo, who had everything ready and was exploring different approaches to get this new gene therapy in babies. MHRA has been very responsive in terms of regulator, and we had a patient who was eligible at the time. So this helped a lot as well to push for getting other sites being set up quickly, and all the teams at Gauche R&D have been very responsive and helpful here.
Amazing. Yes.
Just a quick, I think probably what the question refers to also is the baby KJ, where there was a personalized therapy developed at CHOP, where they managed to diagnose the patient, and then within 6-7 months, there was the first therapy specifically for that baby. This was amazing to hear, and we would like to have a similar approach in a personalized way when you do gene editing, but this is not mutation-specific treatment. So that would be for everyone.
Thank you. And thank you to our speakers, a fascinating topic. Please thank them. Thanks.